cut. I always find it easy to remember this sequence because it's CU, 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 which is the Colorado University repeated 3 times. That was totally by chance, I assure you. And then the intron also provides a binding site for guanosine, which is, or GTP. Either one can work in the reaction, either the triphosphorylated form or just plain guanosine. The important part of the guanosine is that it's hydroxyl group, an OH group, which if this were part of an RNA chain, that OH group would be joined to the next nucleotide in the chain, but in this case it's just a free monomer unit, and there's a site that binds that guanosine on the RNA. And then the guanosine acts, if you're a chemist, it's called a nucleophile, but since you haven't had much chemistry yet, let's just think of it as a little pair of molecular scissors that cuts the RNA chain by a reaction that's called a transesterification reaction. And in that process, the guanosine becomes attached to the end of the intron RNA, and that frees up the exon, the part of the RNA that needs to be spliced, and it now has a hydroxyl group, which used to be, up here, was part of a linkage to the next nucleotide, to the A, but now it's a free OH group, and it can undergo a second transesterification reaction, which is really the reverse, almost the reverse of the first transesterification. Well, normally if you do something forward and then reverse, you're back to where you started. But the reason that this isn't quite the reverse reaction is that there's been a conformational change. There's been a swapping of partners occupying this guanosine binding site. So up here it was a free guanosine from the cell that was bound into this site. Now it's a guanosine that is part of the intron itself. You can see this one that was hanging out there has now bound into the guanosine binding site. So that now when a reaction that's essentially the reversal of the first step takes place, because of this change of occupancy of the guanosine binding site, the reaction goes on to produce the ligated exons. That's the product that the cell cares about. That's now the spliced RNA, and to release the intron as a free molecule, which then in the cell is very rapidly degraded. Now let's look just at a little bit more detail at this process of transesterification. And you can see that the RNA chain this is the exon sequence, the last 2 repeats of the CU, and then this is the beginning of the intron. Here's the guanosine, here's its OH group, and it attacks at a phosphate that joins the U and the A, and then the G becomes joined to the A, and now the U is released. So there are 2 questions that we have to address with respect to this transesterification reaction. How does it occur so quickly, and how is the accuracy or the specificity for a particular site achieved? And I already told you a little bit about the accuracy question, that base pairing interaction helped specify which set of nucleotides would be immediately preceding the site where the RNA would be cleaved. Well, if we want to talk about speed, it might be useful to know how fast would this reaction take place if it was not in a catalytic center. And that can be estimated, the rate of cleavage of an oxygen-phosphorus bond uncatalyzed, and then we've measured the rate of cleavage of the oxygen-phosphorus bond when it's wrapped up inside of the folded structure of the RNA. And the difference between these 2 numbers, this is a logarithmic scale, is 10 to the 11th fold. That's where the 100 billion times comes from in the title of this lecture. So let's put that in terms of real numbers. It means that the reaction, when catalyzed, is taking place on the time scale of milliseconds. So a millisecond would mean it could take place 1,000 times in the blink of an eye. And if it were uncatalyzed, it would take place on the time scale of hundreds of years. So you can clearly see that this amount of catalysis is the difference between the reaction taking place with enough frequency to be very suitable for a biochemical process, for something that has to go on many times per second in a living cell, or if it was taking place once every few hundred years, you wouldn't have a very useful rate of the reaction. So we have to explain then that large rate acceleration. The system we have used is, as time goes on, less and less the intramolecular, within the same molecule, rearrangement seen in self-splicing, and more is an RNA enzyme version. So what we've done is to remove the green portion, the intron, which is the catalytic center, away from the parts of the molecule that are acted upon, shown in blue and purple. This catalytic entity now in solution does nothing. It just sits there, but when provided with another RNA molecule that contains the CU sequence, the two of them can get together by base pairing and now form a structure which is a bimolecular analog of what was occurring within a single molecule in self-splicing. In the presence of guanosine as the cutting molecule or the nucleophile in the reaction, then a reaction that is an analog of the first step of self-splicing takes place. The RNA is cleaved with guanosine attachment, and the catalyst portion is restored in its original form so it can come back and grab another substrate molecule and go through this process multiple times. Why was it so important for us to divide this molecule into two pieces to make an RNA enzyme? Well, if we're interested in the questions such as recognition of substrate by catalyst, it is very much advantageous to be able to change the concentration of the catalyst separately from the concentration of the molecule being acted upon, which we couldn't do in the case of self-splicing. In addition, using chemical synthesis, and I'll show you an example in a few minutes, we can make the substrate molecule by organic chemical synthesis, and instead of putting just standard ribonucleotides at each position, we can put chemically altered bases or sugars or phosphates to ask very specific questions about how do each of those atoms on the molecule contribute to either recognition of the substrate or to the rate of cleavage. And by having this molecule as a smaller molecule, it very greatly facilitates the complete chemical synthesis. Although using more recent techniques, we've actually been able to make single atom substitutions within the large catalyst portion as well. I think it helps to put this catalytic cycle into a structural context that is a little bit more accurate. So here we see a model that was produced by two Frenchmen, Francois Michel and Eric Westhoff, that shows two folded domains of the tetrahymena-ribozyme. The one shown in purple includes, among other features, a binding site for guanosine, and the yellow arrow shows the direction that guanosine attacks. The internal guide sequence is shown as this red strand, and it is base paired by the rules of Watson-Crick base pairing with the RNA substrate, which is shown as the black strand. And you can see that the yellow arrow is pointed to cleave the black strand. And as many proteins have an active site cleft, this RNA enzyme has an active site cleft. This paired region number 1, consisting of the internal guide sequence paired with the RNA that's going to be cleaved, is nestled in a cleft between the purple and the green domains of the RNA catalyst. So then the complete reaction cycle would look a bit like this. It starts out with the substrate as a separate molecule from the